Yeah, not to mention difference in, effective population size and everything. I mean, if they're, well, we'll talk more about it later. I don't buy that for a second, but sorry.
I'll have to get out the article and refresh my memory what exactly is it. That's all I got for now. So we just kind of looked at all these results and decided that evaluating genotypes with microsatellites might assist in monitoring conservation. The morphology that we saw at the lowland versus the mountain populations have a genetic corollary, somewhat. Might be driven by selective pressures that are leading to local adaptation, and we saw, I think pretty clearly, that geographic distance isn't a good predictor of ecological distance for these animals. And then microsatellites were useful for us for describing population structure at a level beyond just morphology surveys. So that's it. Thanks again. I know I mentioned Scott and Veronica a couple times, but they definitely were the drivers behind all of this work. Dr. B, obviously, very much missed, along with Catherine. If any of you remember her, she definitely taught me all that molecular stuff that I know, and other members of Dr. Betancourt's lab were very helpful in my project as well. And then thanks to my other committee members, Dr. Hochberg and Dr. Garb, and then all of the biology department, you've been so supportive and repeatedly asking me when I was going to be done with my thesis. And then, of course, my long-suffering husband and family who kind of quit even asking me about it. That would be a better method to get me to do it. So thank you very much. I told you they were cute. The questions all get asked, or does anyone have any questions?
So I don't know how you guys want to do it, but traditionally, like, questions for the public, and then maybe we'll hang out for a little while afterwards. So I ask good ones now, and or reserve a few for later.
I can just wait till later. There's tons of other
Sure. Yeah, we'll work on it.
I have a question. So how long do they live for?
A long time. I think there's a Petranka paper that said like 16-ish years. Does that sound right, Scott?
I didn't hear the question.
How long is the age of spots?
Yeah, that's a good question. We've been trying to figure that out for a few years, but it seems like I think in captivity they have lived to almost 20 years, and we've done some skeleton chronology where you slice the digits and then cross-section them and stain them with the bone. And I'm pretty sure we've counted some animals that are definitely in their 20s. And probably older than that.
So that might be one of those cases where you have them as a pet, and if you feed them too much, they die quicker, really, than
Yeah.
when they're a little bit more calorie deprived out in the real world.
Because it's just like growth rings,
Yeah.
like a tree. The salamander stops eating and growing in the wintertime, stays nice and cool, and then it has a line of arrested growth right there, so you can tell, but it's still kind of controversial.
So you can keep studying them for a long time if you
Yeah.
want to stay at
You can't
university.
wait. Yep.
The Milford population, we didn't talk too much about that, but that's one of the more interesting ones. And as far as the animals there are consistently smaller than any other population that we found. And you said something about there being a little bit unique as far as the alleles go compared to the other ones.
Yeah, I could pull
How
up.
would you elaborate on that a little bit?
So Milford is that 120 that's on its own. I did mention the private alleles that popped up. Milford was one of the sites that had a private allele at a locus, so it was the only population that had this private allele. And it was the AMA 12-7 allele. So this allele here, I was hoping it was the more interesting one. So this allele here occurred at a frequency of 2.5%. You probably can't even see it, it's so small. But it was the only population that had that allele at all. So that was interesting for Milford. There was another private allele at the Matches Pond, and it occurred really frequently, so that was pretty interesting. But that's not getting at your question of Milford at all. But they are genetically distinct. You can see this they are pretty far away from these two populations, but similar elevation. But the frequency of this allele is so high in these two populations that it kind of looks like it's going away now. So, it's hard to say.
I'd say that the elevation is interesting, but I think that the ecological conditions surrounding these ponds, the terrestrial habitat where the salamanders spend over 90% of the time, is much likely more important than the elevation, even though the elevation has to do with those conditions around there. In other words, at the top of Wachusett, very moist, rich soil and cool in the summer times. Around that site in Milford, which I don't think you've been to, but it has very dry soil, barely any topsoil. And it's had a history of forest fires. It's the opposite of an old growth forest.
Right. Yeah. I did have to confess in my paper that I didn't look at ecological parameters at all, pretty much.
But that would be a nice place to go with this as a baseline
Right.
for any people looking for a project.
Scott's always willing to take someone on and start them on a new project.
I did want to ask you though, how, because you sort of hit on it, but how did these results jive or not with Veronica's findings from mitochondrial?
Well, her results just showed that it was kind of say this distribution, it's probably too general to say this distribution of genetics, but she did show some distinct differences between the Summit and Matches, for example.
So it's consistent in that
Yeah.
way.
Yeah, so I confirmed Veronica's.
How do they do with warmer or colder climates, like South Carolina or southern Canada?
They have a really high distribution. I don't know how cold it needs to get before they stop showing up, but they do survive over the winters by just burrowing.
If it gets warmer, are they more moist, more warm, sort of an incubator? Global warming, that might be fine.
It might be fine, actually. I mean, I was a little freaked out. Oh, well, not freaked out, but I was wondering how they were doing this year when it got real hot, because they come out when the snow starts to melt and it starts to get warm. So I was wondering if they were coming out to breathe and then freezing, because then it got so cold again. So that's a challenge for them. But yeah, there's a chance that they might be fine. But with a permeable membrane like that, they really need to stay moist. So if it got too dry, they'd be SOL.
Yeah, they're usually underground most of their lives, so that's a pretty good place to stay cool and moist.
So they burrow into like little tunnels?
Yes,
And they all kind of hang out together, or do they all go on their own?
that's a good question. It's hard to follow them around.
I mean, like anthills, and enough about anthills to know how they do,
They're
but
not like communal. Like
they're not communal or anything like that.
Only
They're all single.
breeding season. Yeah,
They don't.
They come together for orgies only.
This particular species isn't good at burrowing. They don't dig their own holes. They use small mammal burrows, shrews and mice and voles and critters like that. And then in the wintertime, they go down deeper to get a little further away from the frost. But, yeah.
Thank you.
Why did you call drift not a real evolutionary process?
Well, okay.
I had to call you to task on that one.
It's not a selective pressure in and of itself.
Sure.
So, okay, it's an evolutionary process.
So can drift give rise to meaningful differences between species and populations?
Yeah.
Sure.
definitely.
So then I would say, by definition, it is an important
Okay, I
evolutionary
should have said
process.
a
Yeah.
selection force.
Cool.
When it happens, it's not. Yes.
I was just thinking, so let's pretend, like kind of a pretend scenario, that there's a nuclear threat to Massachusetts. And you're ahead of a conservation program where you can only select the DNA from one population to represent that species. Would be there a particular population, so a DNA repository, and the species is gonna be wiped off the planet, but you need to select a population. Is there a particular population that might best represent the species and future preservation? Again, we're talking about conservation, right? That's the point.
Right, yeah. I
So-
mean, I guess personally, I don't think that there's one.
This is the population with the greatest heterozygosity would be the best, or no? Is there?
Yeah,
Just in
most
one
of them.
case.
So I would say either that one or the other here. But
Please, in the peanut gallery, let her answer.
I guess I would go with this one, because I think overall this has the largest number of alleles. This one you'd have to kind of try to, yeah, you'd almost have to select for that little one to make sure it's maintained, but
Because that might be,
I hope that never happens.
from an ecological perspective, that's the one that's, seems to be at the limit. Like it is, probably because it's so dry, is my hypothesis, it's so dry there that in the summer times, that's where the bottleneck is. The animals get, they have to breed early, so they mature at a smaller size and don't put their energy into growth. But if they were in a release situation where they had plenty of moisture for a couple of generations, would they then grow to full-size salamanders and relax that minimum body size constraint? Would be interesting. So it's,
Yeah, that would be
from a
interesting.
genetic perspective, that you pick that one just on heterozygosity alone, it kind of makes sense ecologically too.
Yeah, I mean, I picked it allele numbers, but
Yeah.
I don't know, I never comes to that.
You hope all you want. but-
So what about for all the pairwise FST values? Can you test whether they're significantly different from zero? Because I
Yeah.
thought that was a common thing that people did. You know, whether to say whether a level of FST is actually meaningful.
Meaningful.
Yeah.
Yeah, I have to pull up my paper, because I didn't include the key in there, but I'm pretty sure that there was a
You did.
statistical test.
Yeah.
So I just remember which
I
one it
guess
was.
I missed that.
Well, I have to check and make sure that.
I don't think it's like explicit in the text, but we can talk about that afterwards.
Okay, yeah, because I have to track it down. I should at least have mentioned that it was done, if I didn't actually put it in there. Which maybe I did.
Well, it looks like people are, well, no, I was just gonna say, if there's no other good questions for Erin, let's thank her again.